because my goal in this talk is to kind of let you know who I am as I go through a lot of the neuroscience and a lot of the places that I've visited and done research in over the course of my career so far. So that started here in Johnson City, Tennessee, where the STAR is. It's up in the upper northeast corner of Tennessee, where I worked with this guy. His name is Russell Brown. He worked in the Department of Psychology at the time, and at East Tennessee State, we didn't have any neuroscientists. We didn't have a neuroscience program, right? Dr. Brown worked in the Department of Psychology, and I started working in his lab as an undergraduate. I was about 20 years old, so that was about 20 years ago now. And I still keep up with Russ to date. So it's good to have the first lesson of the day. It's good to have good mentors in your life. So Dr. Brown has been a good mentor for me. So in Dr. Brown's lab, I did, like I said, I was an undergraduate at the time, and I also stayed on and did a master's degree. A master's degree, typically you don't have to do one in order to get into a PhD program, but you can do one, and I chose to do it here with Dr. Brown. And in Dr. Brown's lab, we were interested in learning, why do people with schizophrenia abuse stimulants at much higher rates? So people with schizophrenia will use a lot of caffeine. They are frequently very heavy smokers, and they often abuse amphetamines and methamphetamines. So how can you study schizophrenia in a rat? We can't really ask a rat if it's hearing voices. We can't ask a rat if it's seeing things that aren't actually in the room. So one way we do this is we watch the rats yawn. And I'll tell you why that's relevant here in a moment. But the way we do that, we know that the drugs that are effective in helping people with schizophrenia, they work by blocking the dopamine D2 receptor. And just about all of them have some sort of function in blocking that receptor. So what if we actually reversed that receptor? What if instead of giving an antagonist, what if we gave an agonist for that receptor? And what if we did it at a critical period of their development? In this case, we gave them the drug quinperol for the first 21 days of their life. It's just an injection that we gave them once a day for 21 days. And we find at the end of that 21 days, and after they've grown up to become adult rats, they yawn more than the animals that received saline. And that may seem a bit silly or may seem like a bit of a stretch, but we do know that people with schizophrenia, one of the key symptoms that they have are orofacial tics. So oral or facial tics, and that includes yawning. So we do think that the yawning that we see induced by quinperol is a good translational measure, and that's a word you'll hear me say a lot, is translational. Translational behavior that we can use to study drug abuse in these animals. We were specifically interested in an area of the brain called the nucleus accumbens. If you've studied addiction neuroscience or reward neuroscience at all, you've heard of the nucleus accumbens already. But it receives, apologize for that, it receives a dense projection from the ventral tegmental area. And this projection is carrying dopamine axons, which all innervate here in the nucleus accumbens. And the nucleus accumbens plays a role in matching rewarding stimuli, such as sugary foods, drugs of abuse. Anything we find rewarding is going to activate dopamine in that region, the nucleus accumbens. And what we find is we take our yawning rats, who we gave the drug quinperol, when they receive a dose of amphetamine, we see a much larger dopamine response in the nucleus accumbens when they receive amphetamine. And keep in mind, they haven't received quinperol since they were 21-year-old rat pups, and now they're adults. So this is a double hit, and this is causing increased levels of dopamine in the nucleus accumbens that we think could explain why some people with schizophrenia abuse stimulants at much higher rates. So that was the project I did for my master's degree. And after my master's degree, I traveled just down the road to Charleston, South Carolina, to work with this guy. His name is Gary Aston Jones. And we were interested in studying how does the brain adapt to a new situation or a new rule. It seems pretty self-explanatory that you can't use the same behavior in every situation, right? You have to be flexible. And that was the specific cognitive ability we were looking at, called cognitive flexibility. And in particular, we were interested in studying this small nucleus of neurons here in the brainstem called the locus ceruleus. And this is how it's spelled here. It's a funny word. And in, I believe, Latin, it actually means blue spot. Ceruleus comes from cerulean, meaning blue, blue spot. And it's called that because in humans, these neurons actually carry a pigment that makes them a very bright blue color. So if you ever saw it in a post-mortem brain section, you would see a bright blue spot right where the locus ceruleus is. And that's how it's got its name. These neurons carry axons all throughout the brain, including up here into the cortex. And this brain looks funny because it's a rat brain. So this is a cross-section long ways, a sagittal section through a rat brain. And we were particularly interested in these projections here that come up here to the what would be the prefrontal cortex in the rat. So we wanted to ask, what is the role of the axons coming from the locus ceruleus up here in the prefrontal cortex in cognitive flexibility? So in order to study this, we taught our rats a game. And this game was played in what we would call an operant chamber. That's another term you may have heard before studying neuroscience. But it's called this because there are lots of things in the chamber that the animal can operate on. In this case, there were levers, and this is the front wall of the operant chamber. In this chamber, there are two cue lights. Both are positioned over a retractable lever. Here, the levers are pulled in. They aren't sticking out into the chamber. And in the middle, there's a receptacle here where they can retrieve a sugary reward when they do the when they make the right choice. They do the right behavior. So at the beginning of each trial, everything is turned off and the levers are retracted. This is called the intertrial interval, or the interval between trials. That is 10 seconds long. So they wait for 10 seconds and then one of the cue lights will come on. Now, on any trial, it can be either of the cue lights, right? It's a 50-50 chance. It's randomly presented. It can be either one. And that light is shown to them for 3 seconds before the levers pop out, right? And so now the rat has to press one of the two levers in order to get the sugary reward. If it picks the right lever, the sugary reward is dispensed here into the fluid receptacle and they can retrieve it. If they choose incorrectly, though, everything in the chamber turns off and they have to wait an additional 10 seconds before a new trial will start. So in this case, they have to wait 20 seconds before the new trial starts. And 10 seconds doesn't seem long to us, but for a rat, that is very frustrating for them. 10 seconds is a long time to wait. So how does the rat know, how do they learn which lever to press? So here there are two rules that they could follow in order to discriminate the correct choice. And we would also call them dimensions. They could completely ignore the cue light altogether, right? So say the rule was only press the left lever. So the cue light's going to be going back and forth, left and right on different trials. They can ignore the cue light altogether and only press the left lever. Or the rule could be press the lever that is presented under the cue light, right? And this is actually the rule we teach them first. So when this cue light here on the right side comes on, press that lever and you'll get your sugary reward. they get very, very good at this rule, right? We train it over multiple days, and we train it to a criterion of 20, a streak of 20 correct trials. So they can do 20 out of 20 trials correct. That takes a few days for them to train that well. So when they do that, we'll put them back in their cage for the night and bring them back the next day. And when we start the game again the next day, we have not changed the rule yet. So it starts off and they're pressing the lever under the cue light again. But then on the 21st trial, we change the rule on them. We change the dimension. So now instead of using the cue light, they're told that they are only going to get the reward if they press the left lever on a trial, right? And then they have to figure this out by trial and error. So suddenly they've been getting 100% of trials correct when they press the lever under the cue light, but now they're only getting 50% of those trials correct. So they need to perform what's called an extra dimensional shift. And this is the big key word in this sort of task. We want them to shift, we want them to be cognitively flexible, to shift to an entirely new dimension that they've not used in this problem-solving task before. And that's our measure of cognitive flexibility. So what we wanted to know is, if we could come out here and stimulate these axons coming from the locus ceruleus right when we change that rule, could we make them better at this task? And we used a technique called dread receptors. You may have heard of them before, but in short, they are receptors that aren't activated naturally by anything in the body, but we can inject a drug called clozapine N oxide, which would typically be inert. But this inert drug can bind the dread receptors out here in the prefrontal cortex and stimulate the release of norepinephrine from these neurons coming from the locus ceruleus. And when we do that, we actually can decrease the number of trials it takes the rats to learn this new rule, right? So simply by stimulating those neurons coming from the locus ceruleus, we made them perform this task better. We made them more cognitively flexible on this game. That was my project that I did for my PhD in neuroscience. And so after that, I traveled all the way cross country to San Diego, California, almost your neck of the woods, a little far south, to University of California, San Diego, where I worked with this guy, Dr. Jared Young. And with Dr. Young, I did a postdoc. If you're not sure what a postdoc is, you can kind of think of it like a medical residency. So I already had my PhD in neuroscience. So a postdoc is like additional training you would do in order to develop a specialty. And my specialty was in translational psychiatry. There's that word again, translational. So translational psychiatry, we were interested in finding good ways to study psychiatric illnesses in animals. And the specific project we were working on was to try and figure out why some people experience depression or mania at specific times of the year. So there's a very well-known phenomenon in bipolar disorder in particular, where patients with bipolar disorder will exhibit more symptoms of mania in the spring and summertime. So this would be hyperactivity. They may be very impulsive. There are lots of behaviors that go along with mania, but they're more frequent in the spring and summertime. But there are more symptoms of depression in the fall and winter time. And this is all on a continuum, right? These patients can still experience mania in the winter time. It's just less frequent, right? It's more frequent in the spring and summertime. And we think there's good evidence to suggest one of the key mechanisms that is driving these different behaviors between mania and depression is the amount of sunlight that they're being exposed to. Of course, in the spring and summertime, the days are longer and they're exposed to more sunlight. In the fall and winter, the days are shorter, especially here in Pittsburgh. I think some of our days, it felt like we had about 4 hours of sun. In that case, some people with bipolar disorder would experience more depression symptoms during the fall and winter. So how would we mimic this in a mouse? An important thing to remember is mice are nocturnal, right? So they're more active at night, where we are more active during the daytime. So for us, where we have longer periods of light in the spring and summertime, a mouse is actually more active in the fall and winter when the days are shorter and the nights are longer. So they are more active during this period. So what we did is we exposed them to different, we call them photoperiod conditions. And to do that, we built these custom chambers. You can see they're very well ventilated, big chambers with these LED lights that we could have full control over. And they live in here in their home cages with other mice in their home cages as well. And the two conditions I'm going to tell you about today are short active photoperiod conditions. So they have a very short active period. In this case, that meant 5 hours of darkness with 19 hours of light over the course of a 24-hour day. And our hypothesis was that this condition was going to cause more depression-relevant behaviors. And then we could compare this to a control group that had a balanced 12-hour light-dark cycle. So this comes back to the question again, how do you study depression-like behaviors in a mouse? And this comes back to a very, very old and well-used task. It's been around for decades now, called the antidepressant swim task. And the way this task works is you get a very large beaker. It's large enough so the animal cannot touch the bottom and it cannot escape out of the top. And we place the mouse in here and it will swim. The mouse isn't in any danger. We're watching it very closely. And when they stop swimming, they actually float naturally. And this is what we're looking for, is the amount of time that they will spend swimming before they stop. And the reason that this behavior is important is that antidepressants, such as the SSRIs you've probably heard of, or most other antidepressant drugs, will decrease the amount of time that the mouse will spend immobile in the forced swim task. So our idea is that if we housed our mice for 2 weeks under this short active photoperiod conditions, we would probably increase the amount of time they spent swimming, that they spent immobile, rather, in this task. So what happens? There's a very interesting phenomenon that was found in these sorts of experiments. And this was actually done by a collaborator of ours, where he was looking in a part of the brain, it's almost right smack in the middle of the brain, called the paraventricular hypothalamus. And in the balanced 12-hour light condition, in this area of the brain, what I'm showing you here are neurons, and the colors are specific to the types of protein that the neurons are expressing. And here, the neurons in green are expressing tyrosine hydroxylase. This is the enzyme that produces dopamine and norepinephrine that I've already told you about. There's another population of neurons here stained in red, and they are expressing a protein called somatostatin. Now, somatostatin I haven't told you about, but it's involved in regulating another neurotransmitter called acetylcholine. And I'll tell you about what acetylcholine is doing here in a moment. So this is under balanced conditions, and you get about a 50-50 split in the expression between these two types of neurons. But when you put them into a short, active winter-like condition, you see a lot more red neurons, right? And what was cool is that these neurons are actually switching their genetic phenotype. So the phenotype meaning what type of protein they're expressing. It was long thought that a neuron that was expressing tyrosine hydroxylase was always going to be that type of neuron. But here they found that the neurons were completely switching the proteins that they were expressing. And then if you put the animal into the swim task, they did spend more time immobile in the swim task. So this suggested that the short active photoperiod condition is increasing the amount of depression relevant behaviors in these animals. So I said somatostatin is involved in regulating acetylcholine. And we know that if acetylcholine levels are too high in the hippocampus, that can lead to depression. So we wanted to ask if we could decrease the levels of acetylcholine in the hippocampus, can we block this behavior? So the way we did this is we injected a virus, and this virus expressed a protein called human acetylcholinesterase. If you've studied how enzymes are named, ACE means it's breaking something down. It's metabolizing a molecule. So acetylcholinesterase is breaking down acetylcholine. So our virus here expressed here in green within the hippocampus that's outlined here in blue is carrying this protein that's going to break down acetylcholine. So we're going to be decreasing the levels of acetylcholine that we believe are too high in these animals. And when we do that, we found that the animals who are housed in the short active photoperiod conditions do express less immobility compared to the animals that received the control virus. Strangely though, in our animals that were in the normal photoperiod condition, we saw an increase in the time they spent immobile, which could suggest that they were experiencing more depression relevant behaviors, which actually did not fit with our hypothesis. The first part of our hypothesis was correct, but this was an interesting finding that we still need to follow up on. So that was my main project among many others during my postdoc. And after I finished that project, I went back across the country to where I am now at the University of Pittsburgh to work with Dr. Stacy Rizzo. And I mentioned I'm a project research scientist for what's called Model AD, which is a large group. It's a large consortium of sites between us, Indiana University, University of California, Irvine, as well as Jackson Laboratories in Maine. And we're developing what's called a preclinical testing core because we want to improve the way that we study Alzheimer's disease and test new drugs. So I'm not going to be able to show you a lot of data today. I've been in the lab for a little under 2 years, and we're a brand new lab. It was just starting when I joined the lab, and most of that time we've spent with COVID. So we've been working a lot of the time at home, so we haven't been able to do as much research as I'd hoped for. But our goal is to improve the way that we study Alzheimer's disease and the way we test new drugs. And I mentioned before, I'm from East Tennessee, and I'm also a huge college basketball fan, so you've probably seen this person on the left here, Pat Summitt. She was the longtime head basketball coach of the Lady Vols basketball program. And she died from Alzheimer's disease when she was 65 years old. That's actually very, very young to contract Alzheimer's disease. And it progressed very rapidly in her. And she died at a very young age. Most of the time, Alzheimer's disease will strike in your late 70s, maybe even into your 80s, before you see any severe symptoms. But in her case, she got what's called an early onset form of the disease. Now, those types of the disease are caused by mutations in these three proteins, PSEN1, 2, and APP. Now, if you have these mutations, the chances are extremely high that you are going to get Alzheimer's disease. It's almost certain that if you have mutations in these genes in the wrong spots, you're going to get Alzheimer's disease. But in terms of the mutations that are seen in people with Alzheimer's disease, they're very rare. They're only about 2 to 3% of all cases of Alzheimer's disease. But other gene mutations that are seen in AD, you can see there are quite a few of them. But in the mice that we've used to study Alzheimer's disease, nearly all of them are ones that express these very rare gene mutations that lead to early onset forms of Alzheimer's-like pathology. So the goal in our center is to develop new mice that carry multiple gene mutations. We can combine that with environmental manipulations such as a high-fat diet or a high-sugar diet that we know confers risk for Alzheimer's disease. We can give them exercise wheels to see if increasing their amount of exercise can help get rid of some of the sorts of plaques and the pathology that we see in Alzheimer's disease. And that's our goal, is to make better mouse models that aren't dependent on these very rare gene mutations that cause rare forms of Alzheimer's disease. And so that's all of the data I had to show you here today. And I thank you for the invite to come talk to you. It's great to meet up and coming students in the neurosciences, and I hope to see you at an IBNS meeting someday in the future. And when you see me there, you can say hi and I'll remember who you were. So thank you, and I'll leave any other time to answer questions.
Thank you, Dr. Kopp. I believe we had a few questions in the chat. So the first one is, are there any animals with an increased LC? And I believe he's referring the first portion of your presentation. And I guess you can go ahead and answer that.
Animals that increase LC. So maybe you're asking if it increases levels of norepinephrine. So the LC is actually the name of the brain region. So where those neurons live is called the locus coeruleus, and they express norepinephrine or noradrenaline. It's the same neurotransmitter, but it's called by different names. So the answer is yes. There are animals that that have been produced that do all number of things at this point. So there are animals that you could use that have increased levels of norepinephrine coming from the locus coeruleus. Yeah, but that's not. We used normal laboratory rats in this case.
I'd actually like to branch off of that. How did you genetically modify these rats to express more norepinephrine? Is that how it works? Or is it done for you? Like you just go to like carolina.com and purchase a rat?
You probably could purchase whichever rat you wanted, but so we used, I didn't get into it. It's hard to explain in 15 minutes, but we have time now, so I'd be happy to explain it. We used a technology called DREAD receptors. So DREAD is spelled D-R-E-A-D-D, and that stands for Designer Receptors Exclusively Activated by Designer Drugs. That's the acronym. So the reason it got this funny name is the guy who was trying to invent it had a wager with his boss that if he could make it work, he got to name the receptor. and he got it to work. So he wanted to call it the dread receptor because he thought that was pretty cool. Anyway, so these dread receptors are receptors that they are a form of the acetylcholine receptor, right? So they took the acetylcholine receptor and they mutated it to the point where it doesn't work anymore. So you can inject this receptor using a virus that receptor is expressed, but it doesn't work anymore. Nothing can bind to it. Nothing can activate it. It's just sitting there. But they designed it further so that it can bind a drug called clozapine in oxide. Clozapine in oxide, if you injected it, is completely inert. It doesn't do anything in your body, right? So now you have this receptor that doesn't do anything, but it can bind a drug that doesn't do anything. But when they combine in the correct brain region, it will selectively activate or exclusively activate that receptor. So that's how a dread receptor works. So in this case, the dread receptor we used activates the neurons and causes more norepinephrine to be released. And we could inject it right here in the locus ceruleus, the medial prefrontal cortex specifically. And so then those axons would express the virus, expressing that receptor. And then when we give the drug, even though the drug is all over the brain, it's only binding to and activating receptors here where we put them, if that makes sense. So that's how DREADS work. You should look up, there's probably a lot of articles about it that you could read about called DREAD receptors. They're pretty interesting.
Yeah, that's really cool. That's cool how you can just create a completely new receptor to solve some of the problems, to advance the research.
There's another one called optogenetics that you've probably heard of as well. Optogenetics, it's the same sort of principle. You would inject a virus that's carrying a special receptor for you. But in this case, those receptors are activated by photons of light. So you could express the receptor out here in the prefrontal cortex and then use a very, very small fiber optic and then flash light in that region to activate those receptors. And that's called optogenetics. They're two kind of complementary methods. We did some optogenetics in these animals as well. It didn't work as well as the DREAD receptors though.
Cool. We have two more questions. One is a little bit more general, so I'll ask that at the end. But this question is, in your third study, you demonstrated how light exposure affects mood. Do you think there is a connection with this and increased depression now with quarantine because people are spending more time inside, especially with the teens and youths?
That's a great question. That's a great question. And it remains to be seen. There's going to be a lot of research done during this period of time. You guys are young, so this doesn't seem as crazy to you, perhaps. I'm sure it seems very, very weird, but I'm 40 years old now, and this is the strangest period of time I've ever lived through. I mean, it's really crazy. So people are going to be doing research on this time for a long while. And I think one thing we're going to find is there are going to be very high rates of mental illness during this period of time. I already know that to be true. We're experiencing we're seeing very high rates of admissions in psychiatric hospitals right now, because this is a very, very strange time. And you can imagine if you were already battling a mental illness, how much this change in your day-to-day life can be impacting you. So absolutely. And it goes well beyond just light exposure. People's lives have been completely overturned and upended. You guys haven't even been able to go to school on a regular basis now. So you've been going to school, in Sanyam's case, for 13 years or more, and now you're spending most of your time at home. So that sort of interruption in a routine can be very hard for people to deal with. So yeah, this is going to be a very interesting period of time for studying psychiatric illnesses and mental illnesses.
We actually got one more question, so I'll ask the question that was asked previously. Let me find it. What tips do you have for someone interested in research? So I assume this person wants to go into research later down the line, or perhaps wants to get involved in research in high school. But yeah, tips for someone interested in research.
So in high school, it's a little difficult to get involved, and I think what you're doing now with your club is great. I didn't have anything like that when I was in high school, and I think the fact that you guys have your own neuroscience club is really impressive. So you're already doing something right. But I would say, for now, participate in this club, stay active, and come to the meetings, and try and read as much information as you can. If you're not comfortable reading the primary journal articles, they can be a little hard if you haven't studied a lot of neuroscience already. But there's a lot of neuroscience that you can read about in the popular press. So just read and participate in this group as much as you can. But when you do get to college, I would suggest as early as possible finding a laboratory to work in. That can be pretty intimidating, right? You're going up, you're an 18, 19-year-old kid, and you're going up to a professor who's done research for 30 years, and you want to work in their laboratory. I would encourage you, do not be intimidated by that. If you find somebody on the campus where you're working who's doing research that you are interested in, go ask them if you can volunteer in their lab. Most people who are doing research at undergraduate universities have undergraduates that work in their lab. And a lot of them will run their labs using undergraduate volunteers. They may not be able to pay for a graduate student or a postdoc, so they will have their volunteers help them with the experiments. That's how I got started, and that's how most of the people I know in this field got started. So that would be my second tip. After high school, try and find a lab as early as you can and just get involved in anything, any research that you find interesting. Go ask if you can help. Go ask if you can volunteer. And typically the answer is going to be yes. If they have time and space, they're going to let you pitch in and learn. So. And then after that, it's just work really hard.
And I think our last question will be, what exactly is the research done on Alzheimer's in your current lab?
Sure. I'm doing what I said earlier was I'm working in a preclinical testing core. What that means is we're not doing a lot of new and novel research. What we're trying to do is develop a way to test new drugs, to develop a better way to test new drugs. So preclinical means that we're going to be testing drugs that aren't to the clinical phase yet, right? They're not approved for use in humans. So this is the investigational phase before you would ever actually test a drug in a human being. So the idea is we would ask people from outside of our laboratory to submit drugs to us that we could test for them. We have a number of behavioral tests that we call a behavioral battery that we carry them through. We also do what's called pharmacokinetics, which tells you about how the drug acts in the body and how it's metabolized and eliminated from the body. We do pharmacodynamic assessments. We have a number of tests that we walk each drug through in order to study them. And then, so that's the way we study drugs. And then the way we study Alzheimer's disease goes back to the mouse models that I was talking about earlier. We want to find mouse models of late onset Alzheimer's disease. This is the much more common form of Alzheimer's disease that strikes in your late 70s and 80s and tends to progress very slowly. And there are not good mouse models yet for that type of Alzheimer's-like pathology. And that's what we're trying to develop. So we're still really developing our program and our system at the University of Pittsburgh so far, which is why I don't have any new or novel data to show you. But hopefully in coming years, as we get up to speed and get some of our new mice, we'll have more data to show you then.
Cool. Thank you so much for your time. Is it okay if I close your screen share? Or you could go ahead and do that, and I'll put up the slides for the end of the meeting. Unless anyone has any further questions, but I don't see any in the chat. So if that's the case, if you haven't filled out the sign-in link already, then it's going to be here as well as the first link. That's the most important one. If you want to leave feedback, we have a feedback form. And if you want to officially join the chapter, then the link to the website is there. You can always navigate there by Googling IYNA and then navigating to our page. So yeah, thank you so much for speaking today.
Thank you.
Yeah.
You're welcome.
And for our members, we'll send out an email later for our next meeting. And that will actually be led by Harshal here, because as a senior, I will be graduating. And so it will be our new officer team for the rest of the year.
Thanks, everybody. Bye-bye.
Thank you.